Hello, I'm Riele, and today I would like to share this pastel painting process with you while talking some nonsense. I've had this apotheosis of war kind of idea for over a decade, a well with a helmet instead of a bucket. But since I can't imagine things visually for years, it stayed abstract and without substance. Because I couldn't see the image, I had to construct it. I wanted to make it real and tangible, which raised the high bar for historical authenticity. And since I am very uneducated in both history and material culture, my ideas were quite banal. At first I thought about a Corinthian helmet. It has an interesting and recognizable shape, but I had no idea what wells looked like at the time. Of course, we have archaeological wells, but what's usually left is just evidence of a shaft in the ground, not what stood above it. And although well isn't some high-tech invention, its appearance can vary a lot between cultures and regions. The other issue is that if someone back then lost a bronze helmet, it wouldn't be just lying around. If intact, it would still be used as a helmet. If broken, it would have been melted down right away so as not to waste precious metal. If a helmet were found after centuries in modern times, it would end up in a curiosity cabinet or a museum. With iron helmets, it's even worse. Iron rusts almost instantly. So I needed to move closer to the present, both to have a clearer idea of well designs and to use a mass-produced steel helmet. I decided on a World War II Soviet helmet and borrowed the well design from the Russian North. The puzzle was complete and I set off painting. This issue of historicity brings me to a book I recently read, or rather listened to while running, The Kokotsky Enigma by Lyudmila Ulitskaya. There is a character, Ilya Iosifovich Goldberg, a geneticist, speaking in the early 1960s, who said, "Let's assume the human genome consists of about 100,000 genes. That sounds plausible." And that number broke my immersion in the story. After my run, I went to double-check the literature. Why? Because the 100,000 genes figure comes from the preliminary estimates of the Human Genome Project in the early 90s. What were the estimates in 1960s? They were all over the place. In 1964, F. Vogel calculated an insane number, 6.7 million genes. He did this by taking the total amount of DNA in a nucleus and dividing it by the length of the coding region of the hemoglobin gene, assuming genes don't overlap and are tightly packed. Vogel himself admitted, "This estimate is disturbingly high. A different line of reasoning might be appropriate." So he tried a different approach, looking at polytene chromosomes and diptera. These are found in the salivary glands of fruit flies and other species, though similar phenomena occasionally occur in mammalian trophoblasts and even in plants. These chromosomes are visible even when cells aren't dividing because each one consists of multiple copies of DNA bound together. They also appear banded. Some regions are more densely packed than others. Each band corresponds to a protein synthesizing unit, and there are roughly 50,000 base pairs per band. If you treat that as your average gene size and scale up to the human genome, you get about 60,000 genes, a far more reasonable number. Vogel also noted that regulatory sequences might take up more space than the structural parts of the genes. That was 1964. But the dialogue in the Kokotsky Enigma takes place around 1961. So what estimates existed before Vogel? Here's where it gets interesting. J. Muller, in his famous paper Our Load of Mutations, approached it differently. He estimated the number of essential loci in humans based on the frequency of harmful mutations. His reasoning was simple. If you had too many deleterious mutations per genome per generation, we'd go extinct. Specifically, there is an upper bound on the number of harmful mutations per gamete. Knowing the frequency of many Mendelian phenotypes in the population, we can estimate how many loci those mutations might hit. That sets an upper bound on the number of vital loci. Muller's estimate came out to around 20,000 genes, surprisingly close to what we know today. By the way, he had a crazy life. He worked under Morgan. After a suicide attempt in 1932, he chose the best place to start from scratch for a Jew, that is Germany, barely escaped the Nazis, then went to USSR and saw Lysenkoism develop, barely escaped again, and got a Nobel Prize for X-ray-induced mutagenesis in 1946. I encourage you to read about him and give this video a like or a dislike for the Nazis and Lysenko. So, would 100,000 genes sound anachronistic for a scientist in 1960? Not really. Why did it feel out of place? Why did I feel it was out of place? Because Ulitskaya wrote this book in the 1990s, and that number, 100,000, is exactly at home in that decade, not in the 60s. In a book written today, I wouldn't have been bothered at all. I would have totally accepted a 1960s geneticist throwing a number of 100,000 genes. But here it feels like an anachronism that leaked in from when the book was written. It's an interesting example of the Tiffany effect, when something historically accurate in a work of fiction seems like an anachronism. The name comes from the given name Tiffany, which might sound out of place in a story set in the early modern times, but was already attested in France and England in the 1200s. It derives from the Greek theophania and traditionally was given to girls born on the feast of the Epiphany. But in this case, the Tiffany effect seems three-way. It involves the historical reality of the depicted time, our present associations, and the time when the novel was written. Maybe I'm being too harsh and maybe my bar was too high, but it is that high because Ulitskaya has a very precise, almost documentary style and actually lived through the period. If she hadn't been born in 1943 or hadn't graduated from the genetics department of the biology faculty in 1968, I probably wouldn't have noticed. Of course, in the 1990s, there was no Google Scholar and barely any digitized articles. You had to know exactly what you were looking for and then go hunt it down in a physical library. And paradoxically, if Ulitskaya had quoted Muller's historically accurate 20,000 gene estimate instead, it wouldn't have aged well by now. Do I recommend the book overall? Definitely. I enjoyed the first part immensely. Ulitskaya doesn't shy away from physiological detail and presents them with a great taste. However, the book could use some developmental editing. The composition feels uneven. But who am I to criticize the lack of realism in someone else's work if you see what my well looks like now? Maybe in 15 more years I can finally do this idea justice in another attempt. Comment, share, and come back for more nonsense. Bye.